Amar would like to know, for bispecific antibody purification, what are the major downstream challenges and scale-up issues? There were a couple of downstream questions here, so why don't we talk about that for a little bit? Yeah, so we'll start there. The major downstream challenges and scale-up issues, and then there's another more specific question that came in from George that I'll hit you with after that.
Sure. I think one of the aspects that I have seen with regards to the different modalities, right, other than naked antibodies, is the impact of the matrix on the steps that we have, right? And that matrix input quality leads to actually different concentration of HCP DNAs and aggregates that impacts the primary capture steps later on. Because of the different components of the matrix, it leads to, the first one can lead to a clogging of a guard filter if in place, overpressure of the capture step. On the subsequent step on the wire inactivation can lead to large amount of precipitates formation. And separation of that obviously requires some creativity. And again, that's something that you may not very easily see at a smaller scale, and you really have to test that at scale. For the polishing steps, I've seen more often the question of the profiling, achieving the targeted purity and removal for the both steps. For example, if you take a KEX step to deliver that. For the UF step, I've seen a lot that especially for the highly potent products that you need a small amount of them, the challenge of actually reaching to the lower volumes on the ultrafiltration, and subsequently with the filling step, with the bulk formulation and filtration part. So those are some of the aspects that I have seen impacting the final delivery of the product for different modalities.
Yeah. Thanks, Amir. I'm gonna hit you with that other downstream question real quick from George. He's asking for a discussion of, and you may have hit on this a little bit in your response just now. He's asking for a discussion of a downstream unit operation in terms of what the step is to accomplish, for example, purification, in terms of CPP and QRA, and risk to the product, for example, microbial or thermal degradation.
That's a very good question. So if I understand the question, so it's a question of basically the unit operations and their contributions on the CPP and quality risk assessment with regards to them on the process. I think one of the aspects that we are also indeed dealing with is basically product instabilities. And that impacts, we will see it actually in the hold times. And often the hold times that we have is something that you do not have the chance to actually at development scale to experience that. People come with a diversity of solutions for it. One of the common solutions that people think of is basically to apply ideas for more like a continuous or intensified capture step in order to actually lower the hold time that we see, especially on the front end. So I think it's very common, for example, that we see these specific enzyme that thyroidoxin, right, that is actually generated during the cell culture step, and the hold of the step at the CCCF leads to disulfide, irreversible disulfide bond reductions. And that's one of the aspects that actually people come with a lot of solutions. I think it's a pattern of Biogen or Genentech to consider aeration for those cultures. At the same time, there are a good number of companies that are considering more Conti processing solutions, for example, Chromacon or BioSMBs, right? To actually try to address this. So going back to the question that, well, when we look at the bispecifics, what are the CPPs that we need to be aware of? There are a lot of them, but I think what I have seen that is impacting our process is actually one of the most obvious one is actually the hold times. And what can we do actually to avoid, to reduce that, to actually process the material faster, and in that way reduce the impact on the final product.
Amir, you're doing a fantastic job fielding these questions. They're very specific questions, and Amar from our audience just reminded me that maybe we should zoom out just a little bit. And the question that he's posing is one that I had for you anyway, a little bit higher level. He says, I think maybe zooming out a bit, what does a typical downstream process development for bispecific antibodies look like? How is it different from monoclonal antibodies?
Well, to my understanding, the type of unit operations that you actually utilize there is not drastically different than a classic mAb. I think on the other side, the nuances and the adjustment that we actually apply to them are the ones that actually are making the difference. And those are the parts that I think, with the intention of reducing the end-to-end time and the whole time of that, people try to squeeze the process, the unit operations. I mean, traditionally, people used to have 2 UF steps, one at the beginning and one at the end. And the more we see is actually one of the UFs is actually taken out and at the front side. We look at basically more of the, in terms of mode of operations of the chromatography steps, people try to go for connected processes as opposed to the bind and elute modes that they are applying it. So, I've seen also a range of processes that they end up actually having a very large amount of product there. And there, I've seen more continuous VF and formulation, something that is actually being applied in order to deal with the large volume of the intermediates. There is this funny game of puzzle that you, what is the good name for it? I forgot the name. But I mean, your floor level is very limited, and then you end up actually walking around with these intermediate trolleys. So that's why actually that people utilize a lot of these continuous VF, for example, and formulation features there. I think another aspect that when I was reflecting for myself over the period, over the time, what is changed is actually with regards to the process analytical technologies. I mean, when we look at today versus 10, 15 years ago, we were able actually to measure more, measure faster, and take action on those measurements quicker, right? And also measure wider range of concentrations, right? Take an example of, for example, the very basic example of the UV measurement, right? So today we do have, for example, with the use of these Repligen's device, Solo VPE, right? You're able actually to range, you are able to faster get the data. Those data are connected to your processes in terms of GMP and data aspect, which is another important one. Data are more readily available. And at the same time, you're actually fit for a wider range of product, especially for volumetric UV as opposed to the gravimetric. So I think what I see in terms of like the maturity of the process that we are dealing with, I guess a naked antibody, if it is more on a more mature side with a large number of products in the market, I think bispecifics are catching up, and the technologies that are actually used there is inheriting on a very well established technologies for MABs and adding on top with the aspects around digital automation, screening, right? Adding on top with regards to the capabilities to um, uh, generate higher titer products in a shorter period of time. And yeah, with these process analytical technologies, I guess these are the aspects that I guess making the bispecific antibodies contributing to the faster growth and availability of these bispecific antibodies.